Today our talk is about the brain ventricles and CSF spaces. The ventricular system is an interconnecting spaces and channels in the brain which are composed of CSF, the chambers are connected to allow the flow of CSF fluid via multiple foramens. The foramen of Monroe lets the CSF goes from the lateral ventricle to the third ventricle, and the cerebral aqueduct of Sylvius makes the flow from the third ventricle to the fourth ventricle, as shown in the diagram. This is the foramen of Monroe, junction between the lateral ventricle and third ventricle, and this is the third ventricle, and this is the C-shaped lateral ventricle with projections into the frontal lobe by frontal horns and occipital lobes by occipital horns, and into the temporal lobes by temporal horns. And this is the connection between the third ventricle and fourth ventricle by the cerebral aqueduct of Sylvius, and this is the fourth ventricle in the hind brain. As we mentioned, the ventricles of the brain constitute of two lateral ventricles, one third ventricle, and one fourth ventricle, and is as the central canal of the medulla oblongata and spinal cord. This is the frontal aspect of the lateral ventricle, showing the prominence of the frontal horns. The lateral ventricles are separated by the septum pellucidum and do not communicate directly. This is another diagram naming the parts of the lateral ventricle, anteriorly the frontal arterial horns, and the body of the lateral ventricle, the posterior horns and the inferior or temporal horns, and this is the third ventricle, aqueduct, and fourth ventricle. The ventricles contains about 20% of the total adult volume of CSF in the brain, which is about 20 to 25 ml of CSF. The body of the lateral ventricle is surrounded by the cingulate gyrus, and the third ventricle is bounded laterally by the post thalami. The third ventricle is a narrow slit-like cavity or cleft between the two thalami, as we mentioned, and it communicates anteriorly with the lateral ventricle through the interventricular foramen of Monroe, and posteriorly with the fourth ventricle through the aqueduct of Sylvius. This is a coronal projection showing the anatomy of the third ventricle, the thalami on both sides, the floor is the hypothalamus, and the roof is the body of the fornix on both sides. The fourth ventricle is a rhomboid or diamond shaped cavity. It is wide and flattened space located just anterior to the cerebellum. This is the fourth ventricle. It looks like a diamond shaped cavity. This is the cerebellum posteriorly, and this is the brainstem, mainly the bones and part of the upper medulla anteriorly. This is the illustrative diagram for the anterior and posterior relations of the fourth ventricle. The superior part of the roof of the lateral ventricle constitutes of the superior medullary velum, and the inferior part of the roof is constituted of inferior medullary velum. So the roof and floor of the lateral ventricles are both the superior and inferior medullary villi, respectively. The fourth ventricle communicates with the cisterna magna through the median aperture of medullae, and laterally with the cerebellopontine CSF space through the lateral foramen of Luschka. It extends inferiorly opposite the inferior half of the medulla oblongata, and extends through the spinal cord as the central canal and terminates in the filum terminale and conus medullaris. The central canal is lined with ependyma, but no choroid plexus or share in the production of the CSF is contributed. This is the termination of the CSF cavity through the central canal into the conus medullaris at the lower part of the spine. The next talk is about the constituent of the ventricles, which is the CSF. The CSF is produced by the choroid plexus. It is formed by invagination of the vascular diameter into the ventricular cavity. It becomes highly convoluted and produces a spongy-like appearance. It enters the third and fourth ventricle through their roofs, and the lateral ventricle through the choroid fissure. And its function is to produce the cerebrospinal fluid. What is CSF? It is a clear, colorless fluid produced by the choroid plexus, found in the ventricles of the brain. The pressure of the CSF is kept remarkably constant based on Monroe-Kelly doctrine. Volume of blood and CSF and the brain at any time must be relatively constant. Some parameters of the CSF volume is 130 ml total volume, rate of production 0.5 ml per minute, pressure 60 to 150 milliliter of water, composition protein and glucose and chloride. Just minimal amount of lymphocytes are seen. Function of the CSF as cushioning and protection of the CNS from trauma, and provides mechanical support for the brain. It is a reservoir and assists regulation of the content of the skull, nourishing the CNS, removes metabolites from the CNS, and serves the pathway for pineal secretions to reach the pituitary gland. Sites of formation, the choroid plexus, mostly formed in the lateral ventricle. Some originate from epineural cells binding the ventricle. The movement of the CSF inside the ventricles is controlled by the pulsation of the artery of the choroid plexus and by the aid of the cilia and microvilli of the epineural cells. The CSF is formed in the lateral ventricle and escapes by the foramen of Monroe into the third ventricle, from the third through the aqueduct to the fourth ventricle, and from the fourth ventricle is powered into the subarachnoid spaces around the brain through the medial foramen of Magendie and two lateral foramen of Lucia. This is the diagram of formation and flow of the CSF. The circulation, as we mentioned, of the CSF starts from the production after passage through the interconnected ventricles, then into the CSF as the fourth ventricle escapes the CSF through the foramina. These are the commonest CSF subarachnoid spaces other than the ventricles. We have here the cisterna magna posteriorly and cerebello-medullary cisterns, pontine, prepontine cistern, interpeduncular cistern, chiasmatic cistern, the factors that facilitate the flow of the CSF in the subarachnoid space, the pulsations and movement of the vertebral column, spiration, cupping, changing position, all these share the function of controlling the movement. The absorption of CSF into the dural venous sinuses is the process that occurs through the arachnoid villi, the main sites of absorption. And these are the arachnoid villi, and this is the subarachnoid space. It starts from down. This is the cerebral cortex covered by the pia mater, and this is the subarachnoid space. And these are the arachnoids and the arachnoid villi, and this is the dural sinuses. The CSF, which passes in the subarachnoid space, is powered into the dural sinuses by the arachnoid villi. Another diagram. This is the subarachnoid space containing the CSF after its passage from the ventricles into the surrounding brain. These are the projections of the arachnoid villi, which serves to secrete and escape the CSF into the dural sinuses, and this is the covering bone and skin. Thank you for listening, and hope to see you again in another talk, which will be on the blood circulation and including venous and arterial supply of the brain.